Hello, everyone. I am Jessica Acosta, and for this podcast, I will share the poster I presented in March 2025 in the ASAS Midwest meeting about soluble dietary fiber, insoluble dietary fiber, and total dietary fiber in feed ingredients used in swine diets. I would like to begin by explaining that dietary fiber is defined as the undigestible carbohydrates and lignin fractions of plant feed ingredients. The most complete and representative analysis of fiber is the total dietary fiber, or TDF analysis, which includes the soluble dietary fiber, SDF, and the insoluble dietary fiber, IDF, as I will refer for the rest of the podcast. There is, however, a lack of data for concentrations of SDF, IDF, and TDF in feed ingredients commonly used in diets for pigs. Therefore, we conducted an experiment to determine the insoluble, soluble, and total dietary fiber in feed ingredients with the objective of establish a database for SDF, IDF, and TDF in feed ingredients commonly used in animal nutrition. For this work, we analyzed 794 feed ingredient samples. Several samples of the same feed ingredient were obtained, if possible, and analyzed for total dietary fiber using the ANCOM fiber analyzer that allow us to have an enzymatic digestion to obtain insoluble fiber and then precipitate soluble fiber with 95% ethanol, as described by the method 991.43 of the Association of Official Agricultural Chemists. Samples were analyzed in the Stein Monogastric Nutrition Laboratory at the University of Illinois Urbana-Champaign or in the lab of Trouw Nutrition in Boxmeer, the Netherlands. The ingredient samples were classified in three groups: cereal grains co-products, and oilseeds and oilseed co-products. Analyzed values within each ingredient for determining soluble, insoluble, and total dietary fiber were corrected to 88% dry matter, and the average and standard deviation were calculated per ingredient. Once all data was obtained, we wonder, is the analyzed TDF representative of all compounds that were not analyzed as other nutrients? Therefore, we calculated the total dietary fiber by adding the ash, protein, fat, and starch and subtracting it from the dry matter, using nutrient composition data from the NRC 2012 or the feed ingredient database of the Monogastric Nutrition Laboratory. Then, we test the hypothesis that the difference between the analyzed TDF and the calculated TDF, referred as delta TDF, is equal to 0, and we analyze this data using the PROC mix of SAS using the ingredient type as fixed effect. Moving forward with the results, we observe that the majority of fiber in cereal grains is insoluble fiber. However, cereal grains like barley and rye contain more soluble dietary fiber than other cereal grains, but oats contain the most insoluble and total dietary fiber compared with other cereal grains. Among cereal grains co-product, we observe that there is a wide range in TDF depending on the parts of the grain that is retained after processing. In general, the TDF in cereal grain co-products consists of mainly insoluble dietary fiber, whereas there are low insoluble dietary fiber. Oats hulls contain the most total and insoluble dietary fiber, whereas flours from rice or wheat are products with a high concentration of starch, obtained after the removal of all other components in the cereal grains, resulting in a very little fiber. In oilseeds and oilseeds co-product, the variation in the TDF is due to differences in the composition of the oilseed, the amount of residual oil in the co-products, depending on if solvent extraction or the mechanical press method was used to remove oil, and the amount of hulls added back to the co-product after oil extraction. Among ingredients, soybean hulls have the most insoluble and total dietary fiber, whereas soybean meal contains the least TDF. Here, I bring a summary of the results based on the groups. We observed that cereal grains contain less insoluble dietary fiber than cereal grains co-product and oilseeds co-product. We also observed that the concentration of soluble fiber is similar between cereal grains and cereal grains co-products, but oilseeds and oilseeds co-products contain more soluble fiber. Insoluble and soluble fiber can vary significantly in their physicochemical properties, their fermentation capacity, and the energy contribution to the diet. Therefore, knowing the type of fiber in feed ingredients give us information about how they may act as a fermentable substrate for the gut microbiome present in the intestinal tract of animals. Regarding the delta TDF, we observed that it was not different from zero for cereal grains and cereal grains co-products, but for oilseeds and oilseeds co-products, this is not true, which is likely due to presence of compounds that were not analyzed using the total dietary fiber method. I want to emphasize why is the analysis of TDF so important. It is because it allows us to analyze all nutrients present in the feed ingredients. Here, I bring a summary of the nutritional composition of several feed ingredients. As we see here for barley and corn bran, we were able to characterize all the nutrient composition pretty much to 100%, which is not possible using other fiber analysis such as detergent fiber or crude fiber methods. However, for canola meal and soybean meal, an additional analysis is required to obtain the concentration of sugars and oligosaccharides, compounds that were not analyzed as TDF. But still, the TDF analyzed in canola meal and soybean meal allow us to characterize 100% of the composition, which will not be possible with neutral detergent fiber or crude fiber values were used. In conclusion, the term insoluble, insoluble, and total dietary fiber accounts for the majority of fiber in feed ingredients and provides optimal information about the fiber composition of feed ingredients, which may be used to determine nutritional strategies and promote the health and growth performance of pigs. Likewise, the total dietary fiber analysis in feed ingredients allow us to analyze the nutrients to 100% in cereal grains and cereal grains co-product, but for oilseeds and oilseeds co-product, an additional analysis for sugars and oligosaccharides is needed. However, the information of the type of fiber in feed ingredients can be used to determine their energy value. And most importantly, with this work, we are contributing to increase the feed ingredient composition knowledge with a database that covers more than 90% of the ingredients commonly used in animal diets. With that, I would like to thank the Monogastric Nutrition Laboratory and Trout Nutrition for the support and collaboration for this work. Thank you for listening to this podcast, and please visit our website to keep updated in the research done by the UIUC team at nutrition.nc.illinois.edu.